Welcome. I'm Dr. Sharon Hausman-Cohen, and I'm going to be moderating and contributing to this talk that really is going to be exciting today, given by Dr. Boles, regarding the causes of complex disease, including autism. Just to give you a little background, it is my pleasure to know Dr. Boles. Really, in the world, he is the neurologist, geneticist person, the geneticist that knows most about autism and complex disease, and does a remarkable job of furthering the literature. It's also my pleasure to be here as his colleague because Dr. Boles and I, as you'll hear from our discussion, we do similar work, but opposite work in many ways. And so as you'll hear as we go on, Dr. Boles has extended and furthered the research in autism and complex illness by looking at rare genes and how they contribute and figuring out how to modulate them. And then I am the medical director of IntellxxDNA, which is a genetics or genomics clinical decision support tool that allows physicians and other licensed healthcare professionals to look at the common contributing factors to autism, things like inflammation, detox, but also gene variants that are found in 3 or 5 or 10 or 15% of the population that are not disease causing, but disease contributing. So with that, you'll get to hear a lot of discussion that'll help you further your knowledge of autism and other complex illness. It's my pleasure to turn it over to you now, Dr. Boles.
Well, thank you. I will first introduce myself with the slide here. Oops. I'm mostly a physician. I see patients. Actually, I should have changed this. I'm now in private practice, no longer at Neurabilities. The last slide will go with my email and everything. I've been a geneticist pediatrician for a while. I was 20 years at Children's Hospital Los Angeles. I am also the Chief Medical and Scientific Officer of NeuroNeeds, which is a company that makes products for autism as well as other neurological and functional disorders. And this talk has come to you by NeuroNeeds. We'll talk about some of the products at the end. In the past, I have been the medical director of Cortigen and 4 other companies that are involved in DNA sequencing, which is where I learned really how to use the techniques to look at the entire DNA, the entire genome, and very much in detail. And then I'm a researcher. I was at USC faculty for 20 years, and I still write papers. In fact, I'm going to show you the first one here. We're going to briefly go over this data to show you how this sort of changes the situation and what autism really is and what can be done about it. This paper just came out this January. So this is brand new work. And this slide is very complex, I realize, but it's the colors that matter. The left side is the first 25 patients, and the right side is the last 25 patients. Now, this is 50 patients in a row that had autism. If they had autism, it was all by ADOS or the ADI or something, that they all had autism. But of course, people with autism are complex. They have other things as well, as you can see that in the slide. 22% were female. That's at the pink here. Half of them had moderate intellectual disability or less. So half of them were normal intelligence to just having mild issues. Many of them were nonverbal. Many of them had seizures. More than half of them had some sort of regression. Some of them really met the diagnosis of PANS or PANDAS as well, if not better, than autism. So this is 50 patients in a row seen in a neurology office that had whole genome sequencing, trios, including both parents. So I think that this is pretty representative of what we see in clinical care. It's not necessarily representative of what would be in the population. These are people that their parents brought them in to get sequencing because there were issues. This really does not include what we used to call mild Asperger's, or people that are really medically normal and just have some behavioral traits. This is the data. This is really the slide that shows that first 25 on the left, last 25 on the right. I know, again, that this is impossible to decipher, but it's the color that matters. The yellow is a de novo mutation. Now, this right here is a principal diagnostic variant. It had to meet a very large number of criteria to be a principal diagnostic variant. It had to be extremely rare. It had to be conserved, meaning you can go, all different species has the same amino acid, yet this child is different. It had to be in a gene that's in the literature directly related to autism, et cetera, et cetera. There were a lot of criteria to get here. To get into this category, it really is that genetic variant is believed to be the principal cause of autism in that individual, not just a contributing cause.
And just to clarify, in case anybody is not aware of what the de novo variant means, that means that because they had sequenced both the parents, they knew that this was not an inherited variant that the mother or the father had given to the child, but because of environmental factors, which we'll talk about in a little bit, you can have damage to the DNA and changes in the DNA, and a child can have something in their DNA that was not inherited. And that's what we are talking about here.
So you can see that about 70% of the cases of 50 that I was able to make an exact diagnosis. Now, since this, I've looked at 100 more of the patients of Richard Fry. I've actually looked at 95 of them to date. And the numbers are pretty much the same, even a little bit higher in the de novo. So 70% would able to make a molecular diagnosis. That is the cause that is the largest contributor to disease, but by no means all of it. Most of those were de novos. You can see the yellows. Half of the kids, well, some of them are adults with autism, have a de novo mutation that is extremely highly likely to be the cause of their autism. The odds ratio was actually was 30. So that it is 30 times more likely this is the cause of autism than it is just to be a false positive. I'm not going to go into all this but we did is we looked at other de novos that did not cause variants that were thought to be related to disease. Those were in all genes, but the de novos that are thought to cause disease were almost exclusively in autism genes. The ones that have color are Mendelian, X-linked, recessive, dominant. That's 20% of autism. Many studies have shown numbers in that area. And the whites here have no principal diagnostic variant. I couldn't say that there was a single nucleotide change or CME change or anything like that, that CNV that changed that was likely to be the lion's share of the main one. That doesn't mean I didn't find anything. In this category here, these are all rare variants in autism-related genes, but they just weren't rare enough, or they weren't conserved enough, or the connection wasn't quite as tight. Any and all of these could and likely are related to autism, but they're more the polygenic influence. I like to think of them as disease modifiers. As you can see, some of the ones I didn't find a principal diagnostic one, there were modifier genes found, and even the ones that had de novo generally have modifying ones. So
Yeah.
autism.
So I was curious, Dr. Boles, are the ones that had the de novo gene variants that you found that were pathogenic, were they in general more serious or more severe in their autism ratings?
No, not at all. If you go back and forth between the two of them, there didn't seem to be any trend whatsoever, and certainly nothing statistically significant between the two of them. The ones that have no molecular findings whatsoever, they tend to be the pandas ones, and I think that they're more immunological and less genetic. But those are maybe 10% or so, maybe as much as 15%.
Well,
The de novos are no more or no less likely to have severe autism.
and even on that pans and pandas, when you say it's more immunological, I would say, again, we're having kind of almost a little other side of the coin, that it's immunological, but that still is genetic. It's just more genomic. It's more common variants. There's good evidence in the published literature. And again, that's some of the work that we've done that is in our IntellxxDNA report, that gene variants that contribute to mannose binding lectin, which is the first defense of the immune system, and TNF alpha, which creates autoimmune risk, and things like that contribute. So all
I
of
agree
those.
entirely. Yeah. This type of analysis I did with 50 patients really only has the power to look at rare variants.
Right.
It doesn't in any way say that common variants are not part of this, It doesn't have the power to look at it, so I didn't look at it.
Okay.
And the immunological ones, there's a reason why that child that got a virus that everyone else has got, that child ended up with a lifelong problem and everyone else just had a cold and when it got over it. And there's a most likely, in large part, genetic. This is the last of the really difficult slides I want to show here. There's two things I want to show in this. This is the same data looking at it in another direction. These here, the variant that I found was, first of all, I didn't just use, all of this was at Variantix, the lab Variantix. It was all trio again, whole genome. The Variantix lab, which I think is very good, they had the variant that I say is very likely to be the main cause of autism. That variant was on the report in the ones in green up here, these here. And that variant was not on the report in these down here. And if it was on the report, sometimes the Variantix lab said it was positive. This is the cause of disease or likely positive. Sometimes it said it was uncertain. And sometimes they put it on the report and they say, but we don't think this is the cause of disease. It's likely negative. So if you just got a whole genome sequence, and this is a good lab. If you get it by another lab, a whole genome sequence, it's not going to be any better. You're going to pick up maybe 40%. And if you only look at those that are positive, you're only going to pick up more like 15 to 20%, depending on which ones you include. These down here was because I had a research group and we looked at the entire genome ourselves, that I was able to find that the laboratory did not find. The vast majority of those are in the novel category. They have never been reported before. Now, a lab may have found them before, but they weren't reported in the medical literature. This is the number of cases that's in that laboratories have reported in neurodevelopmental disease in the world. But if it says novel, it may have been seen like 3 times or so, but no one has ever published it that it's related to autism until this publication in January.
This slide is super important for you guys that are listening to realize, because many times you're going to have patients come to you and go, I've already done whole genomic sequencing or whole exome sequencing, so I know it's not genetic. And you can't say that. The field of genetics is so new that unless you have someone like Dr. Boles, and really, if you're going to have someone look at a whole genome sequencing with autism, Dr. Boles is your go-to. Unless Dr. Boles looks at it, or a researcher that is specialized in this area is looking at it, a ton is going to be missed. That is the main point here. And genomics and genetics are just way too new of a field. Getting all this data published, the other point is, I'm so happy that Dr. Boles is publishing. We also are publishing because we're not going to further the science unless we publish it. But these children, they're expensive to raise. They have a lot going on. And so the more information you can get about them, the better off that you're going to be able to have outcomes. Because one of the things which Dr. Boles is going to talk about right now that we do, that both of us do in our respective research, is not only just identify the gene variants, but what do you do to address them? And that is what makes it useful. So I think that I'm going to turn it back over so you can talk a little bit about some of the cations and genes that you found and how you've also figured out how to address them.
Certainly. Both of us are clinicians, and I imagine that in the audience we're clinicians as well. We do a test because we want to help the patient. Yeah, we'd like to know what's going on, but we'd like to know what's going on because we'd like to make a difference. And that's definitely what I do the genetics for. It's not just to identify, but mostly to treat. There are certain pathways that are involved in these genes here. And the pathways are shown in here. It's hard to read this. It's easier to read it here. These are the 7 pathways which are come out of this research. Now, there are other pathways that are important that didn't come out in these 50, like methylation and inflammation and cell migration and cytoskeleton. There are certainly other pathways. But when you look at it, there's maybe 10 to 15 pathways that appear over and over again in autism. These are the 7 that came out of this one here. And of these 7 pathways, 6 of them are treatable and 4 of them are readily treatable. The ones that are in dark green here are quite readily treatable pathways. When I get a mutation in those, quite frankly, it could be the one that's the principal diagnostic variant, or it can be a modifier, or both. I know that these pathways generally are treatable and often the kids get better. And then these here, sometimes I can treat it and sometimes I can't. Sometimes it works and sometimes I don't. But the reason why there's a lot of progressive people out there that saying that autism is treatable is because the pathways that go from the gene to the phenotype, many of those pathways are treatable. And as you can see, a lot of these patients, because they had either a mutation that affected multiple genes or multiple different variants in different genes, there's more than one pathway involved in a lot of people. And so these are and again, these four here are the main treatable pathways. And I think we're going to talk briefly about three of those treatable pathways. We're going to talk about cation transport, mitochondria, and neurotransmission. And just a couple of things. When I say that it mostly made a difference, I have patients put on mitochondrial cocktail basically when I first see them, and many of them come to me on mitochondrial cocktail. So this is not just we found something, we put them on mitochondrial cocktail. They're pretty much on that. The genetics, in about 3/4 of the cases, changed my management. That's something I did not do before. 60% I added a supplement that I did not do before, and just under half I added a prescription medication that I did not do before. And very infrequently, it means that you have to refer to another doctor. You're not opening Pandora's box and you find 1,000 variants that you need to send to other doctors. In fact, by far the most of them went to immunology because either A was an immunological gene and I want the immunologist to opine about workup and treatment, or I didn't find anything and I'm thinking that that might be a way to go. And then
Let's
off target. I'm not gonna.
let's
I
skip
wanna-
this slide because of our time and go on and talk
Yeah.
about the
Okay.
pick one topic like the cation or something so we can have time to talk about environmental as well.
I just want to mention off target almost never. 4% bad news. I mean, it's not like you're going to give them a whole bunch of bad news very frequently. In this one right here, I'm not going to go into this in detail, but cations and mitochondria are related. Everything in biology is related to everything else. Cations is a major area in autism. I see cation mutations in autism extremely frequently, maybe 40% or so. They're very easy to treat if you know what the cation is and what to do. Not every cation can be treated and not every patient with a treatable one can get better. But the basic thing with the cations is that you have leakage of positively charged ion into the cell. In other words, the mutation causes the protein, the channel, to leak. There are some other mechanisms we can't get into, but that's the major one.
And,
Positively charged cells are excited cells. If it's a positively charged cell, it does what it's going to do. So what you're doing is you're making all cells hyperexcitable, particularly in the nervous system. And there are many different therapies that can be used to counteract that. And many of them are treatments that autism doctors that are proactive in their treatment are already using. In particular, the mitochondrial targeted therapies, because hyperactive cells run out of energy because they require a lot of energy, and it takes a lot of energy to pump all that back. If you think about like a dam that has a leak in it, if you have good pumps, sure, you can pump all the water back to the other side, but that's going to run out of electricity really fast. You're going to have an energy deficiency. So mitochondrial targeted therapies. Calcium is the main one. Potassium chases calcium out of cells. Magnesium also helps to antagonize calcium. And then there's just the GABA acting agents, in general help in inhibitory neurons. And then glutamate acting, glutamate is an excitatory neurotransmitter, so you want to antagonize that. So there are many things that can help GABA neurons and then antagonize glutamate, NMDA neurons in particular. So these are some of the things. Serotonin is mostly an inhibitory neuron. 5-HTP helps make serotonin. And then in the medications, these are all medications that actually work on the same pathways as here. And many of those are very frequently used in autism, and it probably has to do with the cation. So there's two things to learn from this slide, and I think that we're going to go over this again. One, if you know the genetics, you know which treatments to use. And number two, if you don't know the genetics, not everyone can get that, not everyone can afford that. We know that cations are important at least half the time, and these are the type of things that you should consider, particularly if you have somebody who's hyperactive. ADHD, autism, maybe seizures, maybe migraine, anxiety, hyperactivity type things.
one of the kind of fun facts about some of these calcium channels is one of the more common calcium channels CACNAs is associated with EMF sensitivity. So we have that in our IntellxDNA neurodevelopmental report because it contributes to both autism as EMF sensitivity, and it also has been associated with increased risk of depression. You're also going to find some of these like GABA related activation channels. The reason they respond to the bumetanide, that's another fun fact, is sometimes children with autism, they don't convert their GABA into a calming neurotransmitter. In utero, GABA is an excitatory neurotransmitter. And again, due to genetics, sometimes they're not converting it to inhibitory, and the bumetanide actually forces that conversion on a genetic level. So really interesting. As you start to learn, I think the most important thing is so much of autism is treatable when you start to understand the root causes.
Here is the mitochondrial slide from the NeuroNeeds.com. If you go to the you can find this under the autism label. It's the conditions under autism. The thing about it is that if you click on each of these, it will give you the evidence in the literature that shows that either A, that nutritional component is deficient in autism, or B, that if you supplement it, that there's a study that shows that it improves, or both. All of these here have been shown to benefit in autism and or to be deficient. And you can click to get the evidence. These are frequently used as part of the mitochondrial cocktail to help energy metabolism in general, but they haven't been studied specifically in autism. They're not negative data, it's just nobody studied it, and probably they would work because all of these work. Mitochondrial dysfunction is commonly seen in the genetics, but even in patients in which I don't see mitochondrial dysfunction in the genetics, at least for a mild, it may be a common mutation, a common variant,
Yes,
but it's
there's a
not
lot
a
of common ones that
yeah.
relate.
But even if you don't see it, the vast majority, I would say over 90% of the cases of autism that I do a mito swab on show up mitochondrial dysfunction. And it's usually the same pattern that you see in the mito swab. So it's not just anything being abnormal, but it's really a particular pattern which is common for autism. And this is just to show you that energy is made on an assembly line. This is what you learn in school here. This is what it really looks like. This is a false color electron micrograph of the electron transport chain and the Krebs cycle. It's an assembly line. Electrons go in one end and they come out the other end. If you gave this guy his vitamins so he put his part in cars faster, you're not going to make cars any faster. You're going to make chaos, if anything. Same thing if you energize this machine here. Giving huge amounts of one chemical and hoping to make the mitochondria work better really causes side effects, and it doesn't help as much. And that's certainly what we all used to do decades ago. My approach is to take the entire line and to use 30, 40, 50 different nutritional substances at moderate doses and give all of these guys their vitamins and to try to push it and that seems to work, and it's far more, has much less side effects. So before we go into these, I thought that let's go back to the outline. Actually, there's one thing that I do want to show. Sorry about this. A lot of people don't want to believe that autism is genetic, but it is. Studies have shown 70 to 90%. In fact, most of the meta-analysis that have been done lately are showing 80 to 90% autism. You could say, well, that's 10 to 20% environment, but the environment causes the mutations. We'll get to that. And the environment is very much involved in the expression of all of these mutations. I think every kid with autism has a genetic component that predispose why them, and everyone has an environmental component. Why did it show up then? Why did it show up then? And how can you treat it? So we can't get around the fact that there's a genetic component. This line here is equal monozygotic and dizygotic twins. So this line says it's all environmental. The further you get from the line, the more genetic it is. And you can see some things like Parkinson's and Crohn's and coronary artery disease are almost entirely environmental. And then like trisomy 21 and Rett syndrome are entirely genetic. Well, autism and Tourette's are over here. Autism being by far the most genetic of all common conditions, meaning more than 1% of the population. Tourette's, fortunately, is not common, though it's not that rare either. So autism, of all the common conditions there are, is the most genetic. That does not mean that it's not treatable. And that does not mean that what you have is what you get by any means. And there are a lot of environmental components that are involved in it.
And
So
I
after,
can, I would love to kind of just jump in here for a second on that genetic. I agree that 80% of autism is genetic, but I think you have to expand the definition of genetics and genomics in that we are finding that, yes, there can be pathogenic variants, but understanding, which ties in well with what we're next going to talk about in the environment as well, understanding the contributing factors really is important, because when you look at the way that autism has increased from being 1 out of many hundred to 1,000 children over to 1 out of every 35 boys in one generation, yes, there are de novo mutations. We have messed up our world, and that can cause harm to the DNA, and that's gone up. But also, we are finding even us, and of course, the population of the doctors that are using the IntellxDNA and the research, they're not necessarily seeing as complicated as perhaps a tertiary specialty center like Dr. Fry. But we're seeing that just by addressing the common variants that are associated with autism, so things that, again, can relate to mitochondrial transport, so it's like some of those solute carriers, but that are maybe found in even 10 or 20% of the population, different things that relate to inability to remove pesticides, things that relate to the ability to synthesize CoQ10, all kinds of things relate to the GABA, and that addressing those helps as well. So I think the reason I'm saying this is it's important for parents, even if you don't find something on whole genomic sequencing, or you find something on whole genomic sequencing and Dr. Boles tells you how to modulate it, I'm going to encourage you to go as broad as you can. The great thing about neuronutrients is, again, there is an investment, whether you're doing IntellxDNA or you're working with Dr. Boles, it's a great base no matter what, because it's common to have problems with the B12, with the zinc, with the mitochondria, and all these different things. But sometimes you're going to be able to create a map and go, maybe this person needs more or less of this. So I just think it's important to, even if you find something in whole genomic sequencing, as to highlight the fact what Dr. Boles was saying, look for those modifying causes as well, or think about them. I had one child in my practice. I don't take children with severe autism. He had been to every place you can imagine, including a very well-known clinic. And his issues, we got him tremendously better in 2 months, going from having 1 to 3 word kind of yes, no, okay sentences, to being able to have a good emotional IQ and saying, hey, Dad, you shouldn't treat people that way on Valentine's Day, when Dad was a little snipey. And most of his issues were related to inability to synthesize CoQ10, B vitamins, and lots of other things, requirements with vitamin D, all of these things. And neuronutrients, Spectrum NeuroNeeds was really important for him, because he had a gene variant that would be considered common. It was called Sybil, and it makes it so your mitochondria lose their B12. It chews it up faster, essentially, and had other variants relating to his vitamin D synthesis and CoQ10. And so using something like NeuroNeeds and a few other things based on what we found, we got this child better in 2 months compared to what had been done in the 10 years previous. So look at the rare, but also the common.
Yeah, I agree. I mean, the autism is complex. And it's complex not just in that different people have different genes or different phenotypes or different mechanisms, but in each individual, there are different causes. There are de novo causes in at least half of them, and those are not in the parents, and they're based upon mutations that probably occurred in the sperm cells, the spermatogenesis cells of the father, maybe decades before, maybe even early in the child's life when he was a boy. because most de novo mutations occur on the male germline. Some are on the female, some are after conception, but most of them are on the male germline. And that's really scary, is that mutations occurring today may cause autism cases 30 years from now. But there are also rare mutations, and I showed you some of those. There are uncommon mutations. I look at those as well. And then there are common mutations. And where our two approaches overlap is on the uncommon. I look at the extremely rare to the uncommon, you look at the uncommon to the common.
Can
And
we
if
go
you do both of our approaches, you get the whole thing across. Plus, there's environment as well.
Yes, absolutely. And can we go back to the ingredients of NeuroNeeds and talk about how some of the specific ingredients push the detox, push the inflammation, push some of the common nutrient variants, how zinc is a cofactor? Because I think it was really very specifically designed with some of these different the different needs, and how they even help with those with the different cation channels.
Well, these are our major things. I mean, there's a lot of other things NeuroNeeds makes, and they're add-on products for the most part. These are lemon and berry flavored. This is in a powder. I usually prescribe this for my patients that cannot swallow, and Energy Needs very similar, and it has powder in a capsule form. When somewhere around 8, 10 years of age or so, kids can start swallowing capsules, I recommend going over to Energy Needs. It's more adolescent or adult dosing in terms of the percentages of everything. But I'd say that they're 90% equivalent. They include a lot of different cofactors, including some CoQ, but we'll get to that, carnitine, alpha lipoic acid, all 8 B vitamins, vitamin C, D, it's also E and K, and A, and a lot of other things in there. And then minerals, there's many, many different minerals. There's about 10 of them, and then this is just some of them. And then there's things that don't really fit in any of those, or creatine. So there are about 25 to 30 components are a mitochondrial cocktail that help the mitochondria make energy, but also detoxify the mitochondria. It's like a car that's going too fast, going up say, pulling a trailer, going up a mountain. You're asking the car to do too much. It's going to burn that gasoline only halfway. You're going to produce a lot of pollution. That pollution is going to damage the engine. Same thing with a mitochondrial dysfunction, either acquired or genetic or both. You're making a lot of half-burned fats, half-burned carbohydrates. You're making a lot of free radicals. You need to remove those to detoxify the mitochondria. There are many things from glycine to carnitine, and the antioxidants, quercetin, rutin, and many others that are detoxifying mitochondria that aren't working as well. So I recommend to do one of these 3, either for those that can't swallow or those that can swallow. Some adults like to take this, and some kids prefer to have mom flavor it themselves, and so they open the capsule. So you can go either way.
Yeah. And even the environmental things like mercury that we know, mercury and pesticides. So those are all oxidative stressors. So antioxidants like the alpha lipoic acid, they're there very specifically to also address the environmental stressors. Carnitine, if you think about that mitochondrial Krebs cycle, which I think that until you enter the field of autism, you go into medicine and you try not to ever think about mitochondria again, because it's, you go, oh, do I really have to think about mitochondria and, in the basic science? But remember, your mitochondria, your Krebs cycle can only use either pyruvate sugar or carnitine as an energy source. And there's a lot of these carnitine deficiencies are one of the major things that we'll see, or different carnitine transport issues are pretty common in autism. So having extra carnitine can be really helpful. And then again, as I mentioned, one of the common gene variants that in the young man, that my first autism patient that I treated with genomics, he had an inability to synthesize CoQ10, but also an inability to recycle CoQ10 to its UBQH form, which ties into the next slide, in that's a pretty common gene. There is a gene variant called NQ01. It's found in 30% of the population, so extremely common. And then there's much less, I think about 10% of the population that have 2 copies. But 1 copy of that gene decreases your ability to convert CoQ10 into the antioxidant UBQH form by 67%. And 2 copies of that gene decrease your ability by 90-some%. So that's even though, so 10% of the population are already set up for really failure when it comes to their antioxidant. And we have so much more oxidative stress. And then remember, the purpose of mitochondria, one of the simplified purpose is with that electron transport is ultimately they're in charge of getting you and recycling that CoQ10 to the UBQH. So, and feel free to correct me if I'm not explaining it well, but you need that UBQH. So I think that the Q needs ubiquinol is great because you don't have to know, do the people have a genetic variant that makes it hard for them to convert it back to the antioxidant form?
This right here is, you can see these are gel capsules. There is only one company in the world that makes a liquid CoQ. CoQ, the chemical doesn't taste very well, and it's a very expensive product, but it's a good product. Most of my patients, what they do if they can't swallow is they cut the end of this off with scissors. There's an oil inside, and then they mix it. Hershey's chocolate, straight out of the container, the stuff that a few drops make chocolate milk, without dilution, that's what I suggest. But there are many other ways to flavor it as well. And we'll get to the and that's also going to be an issue with your krill oil situation. This is the QH part, or QF, QHCF type of ubiquinol. The coenzyme Q10 that you buy over the counter is almost all ubiquinone. It doesn't even say that, it just says CoQ. And the bioavailability is at least 5 times less than ubiquinol. And this one's probably is higher than most ubiquinols. So the over-the-counter CoQs are really not any good. You're wasting your money. The reason why you need to get this and it's not in the energy needs or in the spectrum needs is because this is an oil. It's a very, very lipid soluble. It's not water soluble at all. It doesn't mix with the powders. I did put some in there to keep people from dying of CoQ deficiency, but that's pretty much all that it'll do. You need to take this in an oil. Powder forms won't work. And then the next one, omega-3s. Omega-3s are made by microorganisms. I'm sure that your physicians, you're aware that omega-3s are extremely important for brain health, for heart health, for skin, hair, nails, blood pressure, blood sugar, everything. Microorganisms make it. If you're a cave person, you're eating dirt and mold and mildew, you're picking things off the ground, you're not washing them off. You're getting a lot of it. If you're a fisherman and you eat everything off the sea, well, the plankton have it as well, and it goes up the food chain. But if you're not eating a lot of seafood, and if you are eating a lot of seafood, you're getting a lot of mercury, you're deficient in omega-3s. So most people, most nutritionists and doctors and everything are recommending omega-3s. Fish oils, what most people are taking. Fish oil is fine for most people, but if you're treating a brain disease, you need to get it across the blood-brain barrier. Fish oil is triglyceride bound. It does not cross the blood-brain barrier. The liver will convert some of it, but only a little bit. So huge amounts of fish oil, you only convert a little. More fish oil, you're still only converting the same amount. If you want to get a lot of omega-3s into the brain, it has to be phospholipid bound. Krill is a natural source of phosphatidylcholine bound omega-3s. So this is a blend of high dose fish oil, which is great for heart. It's enough for the FDA to say that we can label it for heart health, plus krill oil that goes directly into the brain, plus, which is on the back, there's phosphatidylserine that's derived from sunflower seed is also in this product, and that goes directly to the brain. So this is fish plus krill plus sunflower seed. They are capsules, so you have to cut them if you can't swallow and then flavor it. I keep looking. There are no krill products in the entire world market that I can find that are in a liquid form because it tastes like shrimp.
So one of the things for you guys, many of you, I can't see all of you because many of you are hidden, but many of you are probably old enough to remember Viarin, which was an omega-3 attached to phosphatidylserine that used to be available from Israel for ADHD. This is omega-3 attached to phosphatidylserine. So it's not just applicable for autism, it's also really beneficial for ADHD. But I also want to comment on the astaxanthin. So I am a dual board certified in integrative medicine and family medicine, and what I would kind of call is my specialty, because I work in genomics and how to modulate it, is how do different supplements work. Well, astaxanthin is one of the main activators. It's not only a really potent antioxidant, but it's a very potent activator of BDNF. and making sure that you have enough mature BDNF, which is important for synaptic attachment and synaptic plasticity, is also important. Another gene that is associated, again, common, but associated with autism is something called PON1. And PON1 removes all your pesticides. So that makes sense because we know that pesticides are not particularly good for autism. Well, astaxanthin also helps to upregulate. So let's say you have one gene that's not a variant that is decreasing PON1 activity. The astaxanthin that's in the omega needs is going to also help upregulate your PON1 activity. So there's a lot of, this is really very, very different than any other omega-3 out there. It's the only one that has that phosphatidylserine and astaxanthin in it.
The other one to show is we've been talking about cations and mitochondria the entire time. And I just show that cations affect mitochondria affect cations and everything like that. But we didn't really go into neurotransmission, which obviously is important in the neurodevelopmental disorders, is that you not only use cations and mitochondria to power it, to send the impulse down the neuron, but you got to connect it to the next neuron. Autism, in large part, the phenotype of autism is an imbalance between excitatory and inhibitory neurotransmitters. And in most kids with autism, you get more than just the autistic traits themselves, which are overexcitatory, but you also have, in most of them, you have ADHD type problems, you have anxiety. Many of them will have seizures, up to a third of them. Many of them will have migraine, and et cetera, et cetera. They're excitatory things. And so it's the balance between the excitatory and the inhibitory. And that is related. Everything in biology is related because it's the inhibitory neurons are tonic. They're always on. It's like the mother and the cookie jar and the toddler. I want a cookie. No. I want a cookie. No. I want a cookie. No. I want a cookie. Okay. That cookie is gone. When the inhibitory neurons stop firing, the excitatory neuron takes over. The inhibitory neurons are always on. They run out of energy fast. So if you have an energy deficiency situation, which is over 90% of autism, it's your inhibitory neurons that are affected most. That's why people say, well, if my kid doesn't have cellular energy, why does he have too much energy? Because it's the inhibitory ones that are turned off. We need to help that balance a little bit. You can do two things. You can decrease the excitatory neurotransmitters and you can increase the inhibitory neurotransmitters. And I've already gone over some of that. There are many things like memantine and amantadine that are sometimes used as drugs. There are many things that can help with the inhibitory ones as well. And then magnesium and zinc, of course, can do both of these. They decrease this and they increase that. But one of the things that can be done is that you can do this naturally. GABA is the inhibitory neurotransmitter. And GABA itself, some of it actually does get to the synapse when you take it by mouth. And people say that GABA can kind of calm them down a little bit. L-theanine is from plants. It is what makes, it is why people like to take tea. It's not just the caffeine. The L-theanine soothes you, calms you down. It's not, but it's not sedating. What L-theanine does is it increases levels of GABA, but also serotonin as well and dopamine.
It also increases the GABA receptors, which is really exciting because if you give people a GABA product that has, it's all GABA, it can be sedating. But if you're giving somebody theanine upregulates the GABA receptors. So you don't need to give as much GABA because you're going to respond to your natural GABA. And L-theanine has been studied in ADHD studies as well. So there's advantages. It's not just, again, about treating your autism patients. They showed in a group of, it was a Japanese study of Japanese students that were taking a really important exam, that the students that got theanine beforehand scored better. So it's a great thing.
5-HTP, 5-hydroxytryptophan is what it's made into serotonin. It goes to dopamine and then dopamine and then serotonin. And that can be very helpful. It's often used for anxiety, depression, irritability. Serotonin is mostly an inhibitory neurotransmitter. Yeah, you can use an SSRI or an SNRI, and many of my patients are on it and they do great on it, but this is a natural way to get there first. I have a lot of patients on 5-HTP and on an SSRI or an SNRI. I've never seen serotonin syndrome. That happens in rodents, not in humans, unless you overdose. And the 5-HTP is only 100 milligrams per capsule. If you go over if you go just to the Rite Aid or CVS, it'll be 500 milligrams.
The other reason for 5-HTP is the most common genomic variant that contributes to anxiety and depression is something called HTR1A. Don't worry about the name, but it is quite basically a dysregulated autoreceptor for serotonin, or a thermostat. So people think they have more serotonin than they have. And so if you give an SSRI, you're not going to get the same benefit for that child's anxiety that you would get. You're trying to recycle something that doesn't exist. And so the most common contributing gene to anxiety is 2.7 times the risk when you have even one copy, really is about not having enough serotonin because your body thinks you have more. So giving 5-HTP and also vitamin D and the other vitamin D and magnesium are other important precursors that you need to make serotonin, and letting the child make more serotonin, or the adult make more serotonin, makes a lot of sense.
The other thing we talked about, magnesium. Magnesium does it works in at least 300 different enzymes. There's probably another 300 that we don't know about. It also increases GABA and it decreases NMDA. More magnesium is generally very good. And it has almost no side effects except for like diarrhea if you use huge amounts that are used for constipation. And then B6 helps to synthesize GABA and serotonin. So there's B6 in it as well. What's missing from this is zinc, and I usually give zinc separately because a lot of people are on other products that have zinc, including Energy Needs and Spectrum Needs. And so a lot of people are on enough zinc, and we don't want zinc toxicity. So I will add zinc in many cases if I think that the amount of zinc they're on in all of their diet and their supplements is not exactly where I want it to be. But I didn't put zinc into this product because sometimes you don't want to add more zinc.
Dr. Boles, before we go on, Chris has wanted me to remind everyone. Chris is, many of you have met. He is the CEO of NeuroNeeds, and he's bringing us this webinar. He said, if any of you guys have any questions, this is meant to be interactive. Please either you can use the thing to raise your hand, or the best is just to put it into the chat. I'm watching the chat because we would be delighted to answer any questions about autism, genetics, genomics, ingredients that you use to treat different things. And we also at the end will tell you how to access further training on genomics through IntellxDNA, and to access Dr. Boles' services through his practice.
Oh, I see a hand raised, or
I'm not, go ahead if someone,
oh.
I don't see, oh, okay, it says, to what extent is there felt to be at times a causal worsening of core autism symptoms on the final weight-based dose of NeuroNeeds? If so, what might be the mechanisms, and what would be approaches to try on a lower, should we go to a lower dose, discontinue, start? And I think what this person is most likely talking about is, you know how there are some children, you start to give them methylfolate, even folinic acid, and they get more hyper. So how do you handle the children who you give something like NeuroNeeds that's high in the B vitamins and they start to get hyper?
Well, there's the mantra I repeat over and over again: start low, go slow. For everything and everybody, but particularly in kids with autism. So I usually start at about 1/4 of the dose. The dose is written on the container. It's also on the website. It's based on weight. The dose is what I recommend for somebody at that weight. As a physician, you can recommend more, and you can certainly, some people take less because they don't need any more and they do better on less. But I always recommend to go slowly up to the full dose because you might get good benefit with less and even more benefit with more. How quickly do you go up? Well, if the kid is very sensitive to nausea and to hyperactivity, you may want to go up slowly over a month. If your child has no sensitivities, you might want to go up where you increase it every day, by a quarter or so. And so on the 4th day, you're on the full dose. If you have side effects, which are never severe and never non-reversible, I've never seen it, never heard of it, of severe or non-reversible. But over-excitation, which can be at sleep time if you give the dose at bedtime, not sleeping, or nausea can occur. Go down to a lower dose, go up slower. There's a few things that you can also do. There are less B vitamins than energy needs. So if you're using Spectrum Needs, you can go to the energy needs, open the capsules. It's actually a little bit cheaper because it's a month supply for an adult instead of a month supply for a child. It's the basic unit that you buy. And then you can always add the COM needs on top of it. Actually, COM needs was originally made to try to counteract some of the kids that were a little bit hyper when they got this when they got the Spectrum needs. And then we found that the COM needs was actually working great on school test and social anxiety. And a lot of my patients were taking it in the morning to combat that.
And also, do you ever find that sometimes if they're really, really sensitive, first making sure they have adequate B12, because the way the methylation cycle works is if they don't have adequate B12 and then you start giving them all the methylfolate, they may be pushing one pathway, but it affects others. And so sometimes I find if they're super sensitive, isolate an ingredient first for a little while and make sure you get the B, start with a little bit of B12, make sure that that's doing, get the B12 levels ideal, and then try it again. And that has helped in a couple of kids.
The other thing is that what's kind of implied in this is when does it kick in? Well, energy needs is 40 active ingredients, Spectrum needs is 33 active ingredients. They all do different things, and some ingredients like magnesium do many, many different things. They kick in at different times. So I would say that you start to really see major changes at 2 to 3 months. It takes time to detoxify the mitochondria, to heal the brain, and then for the neurons to reconnect in better ways. So it takes some time, give it 2 to 3 months. You will see improvement in the vast majority at 2 to 3 months, and then you will see more improvement at 6 months. It's not full, it's not ready yet. It is something that you have to keep going. Autism is not curable. It's treatable. If you stop it, well, it will slowly go away like it's slowly built up, but sometimes you can see it quickly. The other thing is you said about core autism things. I don't believe at all that core autism phenomena is not treatable. Certainly a lot of the treatment that's working on things like anxiety and pain and nausea are helping the autism phenotype because the kid is less upset. But many of the children, not a majority, but many of the children do have increased language, so significantly increased language skills. The ADHD, which is not a core, that often gets a lot better in these kids. I think that for a large reason, it's because of cation-induced false signaling in the brain, and you get rid of that extraneous signaling and they can pay attention better. But many of the core autism things can get better in some kids or adults. The younger that you start, the better that the results are going to be, and it's variable. Usually, it's when I have the genetics on all my patients, of course, and that's what I do. A lot of it is because there are components that we are using that are working directly on the mutations.
Yeah. So we have another question about thiamine. And I just want to say you mentioned ADHD, anxiety, all of those. And I agree that there's a lot of comorbidity. In fact, when we look at neurobehavioral genomics, we look at OCD, PANDAS, PANS, folate, cerebral folate, genes that contribute to cerebral folate deficiency, as well as ones that, of course, directly contribute to autism. One of the questions, which actually relates to a specific gene as well, but that's not what they're asking about, is about can B1 thiamine as bentophamine or TTFD provide any additional benefits compared to thiamine alone? And I'm going to say one thing about kind of thiamine is a pretty common other gene variant is problems with the reduced folate carrier. And those people, because thiamine uses that carrier, do better with more thiamine in general. But I don't know the specifics of studies in terms of bentophamine or some of the specific kinds. Are you familiar with those? And I'm also going to put something in the chat for people who want to be able to, Dr. Boles put in his slideshow, his publication. I'm going to put one of our publications regarding what we're doing. So if you're interested in more.
Yeah, and there's a lot of thiamine in both Spectrum needs and in energy needs, like 100 times or so the RDA, so that should cover it.
Yeah, they were saying just are there different forms of thiamine that are transported better than others, or that have better outcomes data, and I'm not familiar with studies that have different outcomes data with different forms.
Well, while that's maybe technically true, it's not 100 times better, I don't think.
so that if you have enough
100
iron.
times the RDA or something like that, you're going to overcome it. Yeah.
Okay. And let's go to let's give our slides of contact information, but we'll stay on and take questions because I have a couple more questions. But if we go to the next slide, just that should,
I just want to show this very fast. So this would
okay,
be,
because we have two
and
more questions too.
I know that to add the mitochondrial cocktail, either spectrum needs or energy needs to the CoQ, to the essential fatty acids, consider zinc, consider COM needs. Diet is important, and of course, it's up to the physician. I just want to make sure we got that on the tape. Or get out of jail free card. Yes, I am not your physician for most of you. You need to talk to your physician.
Absolutely. Let's take a couple more questions. We have a question about how long these children end up needing to be on these supplements. When you're counseling parents, do you say your child's going to need this for life? Or do you find that maybe they start on the spectrum needs and the calm needs, but eventually they're able to wean off the calm needs? I can give my thoughts, but I'd love to give your thoughts.
Chronic disease does not generally get better. Autism usually is rather static, unlike many others, but you're not going to you're not going to stop using insulin or blood pressure medications. And most people, you need to keep it on for a long time. Once you get rid of some of the more problematic problems and the kid gets a little bit older and more able to do behavioral approaches, some people can decrease it, but I think that most people find that they do better if they continue it indefinitely.
And what I usually recommend to patients, because every child, every adult can get pill fatigue. First, let's see how we can optimize you and do as many layers of what we do to get you as well as you can get. I also talk about what foods you can use, because there are certain foods that have high, berries have a lot of things in them, and fish in the diet, some of the fatty fish, and doing what I can. But then I say, get optimized using the combination of supplements. Your child you wouldn't be here if your child was doing great. Let's get them well. Don't stop a supplement. If you feel like they're doing great, maybe you go down to only doing it 4 days a week and then see if they're able to maintain the behavior. So I think there's no easy answer as to, I never tell a parent or a patient that's an adult, you're going to be on all of this your whole life. But then I actually saw somebody this week, and she has horrible antioxidant genes. She's in the 2% that has very, very low, something called clotho, which is an antioxidant regulator. She needs to be, and I have her on the adult form, the energy needs, because if she's not, her alpha lipoic acid and all her oxidative markers go crazy. So she needs it for life. So it's going to depend a little on the child. But most of these kids, I agree with Dr. Boles, it's a chronic disease. They're going to need it for life, but you can tell the parents maybe they won't need as much once we get those mitochondria healthier. Mitochondria, when they're damaged, have a unique ability that they can, with fission and fusion, repair themselves. So you do get some repair that then gives benefit beyond that, may not, that maybe that they won't need as much support when they're older, but it just is going to depend on the child. We have another question. If my patient with neurodevelopmental condition tests results show that they are not nutrient deficient, will the mitochondrial cocktail or spectrum needs be helpful? There is no test that is, so first of all, the nutrient levels tested in the urine are going to be for norms. And if you don't, even if you were to do whole genomic sequencing, IntellxxDNA, all the genomics out there, and if we can switch to the next slide so people can learn how to get trained also on IntellxxDNA, you're still not going to know, does this person, for example, have a transporter gene variant that makes it hard for them to transport B12 into their brain? Do they have, again, a gene that makes it harder for them to recycle the CoQ10 into the UBQH? So if you are testing levels in the urine, that's what they're excreting. And it doesn't tell you about the transporters into the tissue or into the brain. Many of these kids have problems with different transporters, cation channels, mitochondria. So I don't really, to be honest, I tend to I do find urine organic acid testing helpful. I don't find all the constant nutrient testing really helpful. I try to gather as much information as I can. Obviously, I use genomics as a big part of that, but I use outcomes a lot more than what a blood level is, because the norms, I mean, think of what the norm is for vitamin D. You can say, oh, this person's got great vitamin D levels, but that doesn't mean it's optimal for brain function.
There's the other thing is that sometimes we're not just trying to reverse a deficiency, but we're trying to push pathways. You remember from college and high school chemistry, A plus B goes to C plus D. If you add a whole bunch of A in there, you're going to have less B, you're going to have more C and D. Sometimes I'm pushing pathways in a particular direction of healing. And so I use a tremendous amount of A because I want to get rid of the B. It's a problem. Or you need more C and D.
Just to go back to, that's what Dr. Boles was talking about with the cation channels, where he's using magnesium he's using magnesium as a drug to inhibit those overactive excitatory sodium channels. So, calcium channels, although it can work for different kinds. So yes, we will, and you can go back to your slide with your contact information, Dr. Boles, make sure everyone has that as well. Please reach out to either of us if you have questions about wanting to incorporate genomics or advanced genetic testing with whole genomic sequencing into what you offer your patients. And the last thing I will say about that is many times doctors are hesitant to talk about something like genomics with their patients or getting whole genomic sequencing done because they're afraid about talking about the cost. A child who has special needs, even mild special needs, even ADHD, let alone autism, will cost their parents upwards of an extra $400,000 to a few $1 million over that child's lifetime. And so we don't want to, if you think something might help you or help that child get better improvement in their symptoms, you don't want to not discuss it with the parent. Give the parent that choice. The parents that I have talked to, what the first thing they said after their child had been optimized using IntellxDNA was like, why didn't I learn about it? Why has nobody told me about this before? It wasn't around 10 years ago. But a lot of doctors, even that know about it, don't want to talk about something because of cost. Give the parents the option, because raising children is expensive. And what each of us want as a parent is that we'll be able to launch our children, that they will be able to go and have a fulfilling life, career. And so every bit of optimization, whether it's with ADHD or autism or anxiety, is going to help that child live their full potential as an adult. And if you are interested in learning more about gene function, you can get free training on IntellxDNA trains on over 400 different gene variants. Not all of them are related to autism, but we do have online training that is free when you if you only order even one report in your lifetime. So if we can go back to the next slide, the last slide again.
I just want to add because we're, oh, okay, you have something
Yeah.
quick. I just want to add because we're running out of time.
Yeah, I was just letting them scan. Please let us know, and we'd be happy to get you trained on that. And again, if you don't have the data, the NeuroNeeds products, they're going to really be a great way to start treating the child, and doing nothing but that alone, you're going to see improvement in kids with autism.
Yeah, a couple of things. I always put the kid on the NeuroNeeds products first. There are other brands out there, but and some of them are pretty similar and everything. But I would really strongly suggest using the multiple approach to treatment of autism by giving 40 to 50 different supplements, and instead of separate supplements, one that gives like 40 in together, the approach that we're talking about here. I do that when I order the DNA or when I first see the patient. You don't need to have the DNA analysis to put them on the NeuroNeeds products. I always put that on first, get the DNA, and then change things around a little bit to around the DNA findings. The other thing is that the variantics is necessary for me to do what I do in terms of the whole genome sequencing to find the de novos and everything, which is a different company. They both give you different results. It's very little overlap between the two. You're getting completely different results from each one. Variantix, it's $2,100 to do a trio. If you don't have insurance, or insurance will pay for it. But I'd say 95% of my kids with autism, insurance is paying entirely for the DNA.
Yeah,
So
and Variantix also is who I use when I need whole genomic sequencing. They do take about 12 weeks to get results back, so don't expect it back tomorrow. And it's variantyx.com. The reason that they are the whole genomic sequencing tool that I think that both Dr. Boles and I like is they're the only tool that gives you access to mitochondrial. So the mitochondria have their own DNA. The mitochondria also have DNA in the human DNA, but the Variantix gives you access to mitochondrial variants as well as the human variants.
And it'll give me access to the entire sequence, and no other lab in the world will give me the entire DNA sequence, only the report.
Yeah,
Variantix will give us the sequence.
They are great to work with. If you want to start digging deep, you can do it, but probably unless you have a lot of time to learn, you're going to want Dr. Boles to do that for you.
Well, I think that we're done now.
Thank you for joining us. We always appreciate you taking the time out of your busy schedules and hope that you learned something interesting.
Learn more at NeuroNeeds.com. Don't forget to like, share and subscribe. Thanks for being part of our journey.